Assalamualaikum and hi to all panels. My name is Wan Nursharah Auni, Binti Wan Zakaria from AS2575C. So my title for FYP1 project is Detection of MAC-A and TET-M gene in coagulase negative Staphylococcus isolates from subclinical bovine mastitis contaminated raw milk sample. Let's jump to the introduction. Subclinical bovine mastitis refers to a stage or condition in a disease where the symptoms are not readily apparent or do not show a level of severity that is noticeable. In the context of subclinical bovine mastitis, the detection often requires laboratory tests. Coagulase negative Staphylococcus cons are bacteria within the Staphylococcus genus, commonly present on the skin of humans and animals. For problem statement, one major issue in the emergence of resistant genes, especially those that result in resistance to antibiotics like tetracycline and penicillin. In subclinical bovine mastitis, preventing and controlling bacterial infection is essential to comprehend the prevalence and distribution of resistant genes. Alright, so research question in my research is what is the penicillin and tetracycline resistant pattern of 3 cons isolates from subclinical bovine mastitis milk samples? And the second research question is do cons showing resistant to penicillin possess MAC-A gene and cons showing resistant to tetracycline possess TET-M? The objective of this research is to perform antibiotic susceptibility testing on cons isolates obtained from subclinical bovine mastitis milk sample, with a focus on the penicillin and tetracycline to confirm their resistance to antibiotics. The second objective is to investigate the presence or absence of the MAC-A and TET-M gene in the cons isolates by PCR. Alright, so the significance of study in this research is to determine the patterns of antibiotic resistance in isolated cons. The result obtained may provide insight about MAC-A and TET-M gene, thus providing information on the molecular basis of resistance. The expected outcomes. This research might confirm the presence of TET-M and MAC-A gene in coagulase negative Staphylococcus isolates. It assesses antibiotic susceptibility, offering insight into resistant mechanism in subclinical mastitis. Literature review: antibiotic resistance. Usually, farmers use antibiotic in animal to treat infections and prevent disease, but using antibiotic a lot can make animal grow faster, which is not always good. When animal get a small amount of antibiotic for a long time, it can make the antibiotic doesn't work well anymore. If we eat product from these animals with antibiotic that doesn't work well, we might get sick. Also, the germs that are not killed by the weak antibiotic can spread to the air, water, and soil. Welcome to the second point. Subclinical mastitis may not show obvious clinical signs. They distinguish by alteration in milk composition via lab diagnosis only. And the last one is increased number of somatic cell count. The last part is MAC-A and TET-M gene. In simple terms, the MAC-A gene carries instruction to make a special protein, PB2A, in bacteria. This protein has a trick. It doesn't get affected much by certain antibiotics which call beta-lactams. Usually these antibiotics stop a part of the bacteria from building, but with PB2A, it's like a bacteria have a shield and they can keep building even with the antibiotic around. While MAC-A is known for causing antibiotic resistance in Staphylococcus aureus, finding it in constraints related to bovine mastitis raises wider concerns, as these resistant traits can make antibiotic less useful for treating infections. In other words, the TET-M gene is like a protective shield for bacteria against a medicine called tetracycline. When bacteria have this gene, they produce a protein called TET-M. When tetracycline tries to work and stop the bacteria, TET-M step in and block it. This helps the bacteria stay strong and doesn't get hurt by the medicine. So it's a bit like the bacteria having a superhero shield to resist the medicine effects. TET-M usually encode ribosomal protection proteins. The TET-M gene is often found in the bacteria and provide resistant to tetracycline antibiotics. In the presence of the tetracycline, the TET-M protein bind to the bacterial ribosome, preventing the antibiotic from inhibiting protein synthesis. The first step is to revive the isolates that have obtained from previous students. The second step is to ensure that the isolates have been revived have resistance toward different types of antibiotic, that is penicillin and also tetracycline. The second step that corresponds with the objective 2 of looking whether there is resistance of MAC-A and TET-M gene in the isolates is to do PCR tests. So it started with the DNA extraction and all the appropriate molecular technique that follows in order for us to get the amplification of TET-M and also MAC-A gene using the primers that I will be shown on the table on the next slides and also the expected size of the bank. Based on this table, TET-M gene forward and reverse sequence with the amplicon size 1070 and MAC-A gene forward and reverse sequence may be involved in these steps to succeed the objective 2. Thank you to all panels for your quality time. At the end of this slide, I also provide a Gantt chart and a reference.